Hello everyone. After we are finished with the structure of RNA, now we are going to the synthesis of RNA. As all of you might be knowing that the synthesis of RNA it's termed as transcription. So we will take up transcription, but before going to transcription in detail, we need to know certain things about this phenomenon. So let's begin. If all of you will be remembering that synthesis of DNA from DNA, it's called as replication. Now, the synthesis of RNA, it takes its message, it takes the directions from the DNA to get synthesized, and the process of synthesizing RNA from DNA, it's termed as transcription. Okay. Now, in transcription, as you know, that there are ribonucleotides of which I've already talked about. In the ribonucleotide, we are having a nitrogenous base. Okay, and the nitrogenous base, it can be adenine, it can be guanine, it can be cytosine, it is uracil, but it can be never thymine. Okay, then we are having ribose sugar of which 2' which is intact, and then we are having phosphate group attached to it. Whole of these, that's nitrogenous base, ribose sugar and phosphate group, they make up the ribonucleotide. And in RNA we are having these ribonucleotides joined together hand in hand by these 3' 5' ends, that is 3' end of one joins to the 5' end of another, forming a long polyribonucleotide chain which we call as RNA. Okay, now the search for these joining, it means that if the bond is formed between the ribonucleotides, there must be some enzyme involved which catalyzes this polymer formation. So search for this enzyme began, and it was Samuel B. West and Gerard Hurwitz in 1960 who discovered an enzyme, and they said that it will be DNA dependent RNA polymerase. Why DNA dependent RNA polymerase? Because this enzyme was able to catalyze the formation of RNA from DNA, taking the message from DNA, taking DNA as a template, so they named it as DNA dependent RNA polymerase. If you will remember, DNA polymerase that uses to take its message from DNA to synthesize new DNA, so that will be named as DNA dependent DNA polymerase. But here we are having the sense of RNA, but it also depends upon DNA to get synthesized, so we will say it's DNA dependent RNA polymerase, and the enzyme was named as RNA polymerase. Okay, since we are studying this rapid transcription in bacteria, in prokaryotes, which is in your syllabus, and in prokaryotes there is just single RNA polymerase, and it's made up of multi-subunits. Like you remember DNA third polymerase, which was also multi-subunit. Likewise, this is also multi-subunit. It's having the units like beta, beta dash, alpha, alpha, and omega. Now, this enzyme, this assembly, it's called as core enzyme. Okay, as such, it is not able to recognize where to start the RNA synthesis. This core enzyme, it just has the polymerization property, that is it's able to join the ribonucleotides, and this assembly we call it core enzyme with polymerization activity. Now, where exactly to start the transcription? It requires another subunit which we call as sigma factor. Sigma factor, it remains associating and dissociating with this core enzyme depending upon its need, where it's needed. This sigma factor is the actual subunit that actually recognizes the DNA sequence as to where the transcription should be started, where the gene to be synthesized is. Then this joining of sigma factor with the RNA polymerase, the enzyme gets completed and we call it as holoenzyme. But remember, this sigma factor, as you will see in the process of transcription, it keeps on associating and dissociating with the core enzyme. Now, each of the subunits in this RNA polymerase in bacteria, in prokaryotes, it has a different function. If we will talk about alpha 2 subunits, They help in the assembly of this core enzyme. They help in the gathering of the subunits together in a right way so as to RNA polymerase is formed. And these are encoded, these subunits, these are protein units, these are encoded by the gene RPOA gene. R stands for ribonucleic polymerase and A stands for alpha. Then come these beta and beta dash units. These are very important subunits. These are termed as a catalytic center and are encoded by the gene RPOB. Now, main work of the enzyme is actually to perform the catalysis, actually to join the ribonucleotides, and that is performed by this enzyme, these two subunits, that's beta and beta dash. More precisely, beta subunit, it forms the phosphodiester bond. So remember this, in RNA polymerase, it's the beta subunit which actually forms the 5' 3' phosphodiester bond between the ribonucleotides. And then we are having beta dash. Beta dash, it binds to the template DNA. Okay, template DNA is the DNA from which message is to be taken as to what ribonucleotides are to be added to form the RNA, and it opens up DNA duplex. If you remember in DNA, to open the DNA duplex, we had helicase enzyme, but here we don't have helicases. The job is performed by the beta dash subunit. It opens the DNA duplex. Now comes another important subunit, that is omega. It helps in the assembly of RNA polymerase, and it's encoded by the gene RPOZ, and its function is to also protect this enzyme from denaturation by working with some chaperones. If temperature is raised and the RNA polymerase, it has fear of losing its 3D structure, and as a result of that losing its catalytic activity, this omega comes into action, and it works in association with certain chaperones and prevents the denaturation of this RNA polymerase up to certain temperatures. Then comes another important subunit which we call as sigma, of which we have already talked about, that it is the subunit which actually recognizes the sequence in the DNA from where the gene is to be transcribed into RNA, and those sequences are called as promoter sequences, of which we will be talking in detail. So its work is to recognize promoters and to initiate transcription, and it's encoded by RPOD gene. Now coming to some important points about transcription. Transcription, you know, it is the synthesis of RNA from DNA. Firstly, this transcription process, it's very selective. When we say selective, it means that whole of the DNA is not transcribed at a time into RNA. Like we had in DNA synthesis, whole of the DNA was synthesized during the S phase of mitosis or meiosis. Whole of the DNA got replicated, but here in RNA it's very selective. Only certain genes which are required at different stages, they are only synthesized. For example, if we will talk about a child, there certain proteins are required in its childhood, and only those genes are transcribed into RNAs which are required in childhood. So when the child begins to grow, those genes which are required in childhood, they are stopped, their transcription is stopped, and new genes which are required in adulthood, they are transcribed into mRNA. This is contrary to this replication, wherein all of the genes are replicated, but here in transcription only selective genes are transcribed, selective genes are transcribed into RNA. Also, if you know that due to certain disease or due to certain conditions like high temperature, different proteins need to be synthesized by the body, and it's so we say the transcription is selective. It does not involve synthesis of all the RNA at a time. And the sum total of all the RNA molecules produced in a cell under a given set of conditions is called as cellular transcriptome, and its study is called as transcriptomics. Now another important point about this is that it resembles replication in its polarity. If you remember, in DNA we had 5 prime and 3 prime end, and the nucleotides they got added only to 3 prime end. Here also in RNA nucleotides are only added to 3 prime end. So the direction of synthesis 5 prime 3 prime, and the addition of nucleotides occurs only on 3 prime ends. Then it also requires a template, as in DNA synthesis you have seen that the daughter strands they take the complementary these. If there is this a parent strand and there is adenine on the parent strand, opposite to it on daughter cell we will have thymine. Okay, fine. Then we have guanine, opposite to it we will be having cytosine. Likewise, if we have here guanine, opposite to it will be cytosine. So one of the strands is required as template. Here also in RNA synthesis DNA is taken as template, means message is encrypted from DNA into RNA, but if we will having, and the complementarity is there. Also, one important thing is that only one strand, since RNA in most of the organisms it's single stranded, so only one strand is used, and that strand from which it takes the message as to what nucleotides are added, that is called as template strand. Transcription does not require any primer to initiate synthesis. As you have seen in replication, DNA polymerases they were not able to add the first nucleotides, and as such there was requirement of a short stretch of RNA, and as a result of that that was named as primer here. But here in transcription, RNA polymerases are able to carry out the transcription, carry out the polymerization right from first nucleotides, and it is usually purine that is added. So first nucleotide in RNA synthesis is usually PPA or PPG. The two complementary DNA strands have different roles in transcription. One strand serves as a template strand and is called as the non-coding strand or template strand or antisense strand. The other strand, complementary to the template and having same sequence as that of RNA formed except for thymine in place of uracil, is called as coding strand, sense strand or non-template strand. Now let's see what it actually means. See, if we are having a DNA sequence with two strands, we are having two strands of DNA. One is 5 prime, 3 prime, and another is, these are the two strands of the DNA. And from this DNA, we need the RNA to get synthesized. Means we need transcription to occur. See, RNA, since it's being single stranded, so it will choose one among these. Which one it chooses, it's a highly regulated process. It depends, is a very highly regulated process, the mechanism of which is too complex, and it still needs to be elucidated that which of these it chooses as a template. Say for example, herein this RNA to be formed, it has chosen this as template. So opposite to thymine, it has added adenine. Opposite to adenine, uracil, cytosine, guanine, cytosine opposite to it. So this strand from which it has taken the message as to what to add, this will be called as template strand. Okay, this will be called as template strand, because from this mRNA has taken the message as to what to add opposite to that of thymine, what to add opposite to that of, so this DNA strand will be called as template strand. Another thing, the other strand, if you will see, which is non-template strand, which has not acted as a template, but if you will see the sequence of this non-template strand and of this RNA, you will see that the sequence is same, except for thymine there is uracil. As an RNA we don't have thymine, and for the rest the sequence is same. So this non-template strand, it's also called as coding strand, because the coding is same in the mRNA formed and in the non-template strand. Also it's called as sense strand, because their sense is same, their nucleotide sequences same. Now this template strand, it's having nucleotides complementary to that of the mRNA. It's called as antisense strand or it's called as non-coding strand. Hope this is clear to you people. Now coming to bacterial promoters. I've already talked to you that sigma subunit is not able to sigma subunit along with the core enzyme is able to find that where as to start the transcription. Transcription is highly selective process and only certain genes need to be transcribed into RNA. So it's always a challenge to find that which gene is to be transcribed. For this gene, it's a functional unit of DNA that can be transcribed into RNA. To the gene, there are adjacent to the gene, there are certain consensus sequences. Consensus sequences are the sequences which are conserved, where we know the sequence of the nucleotides. To this gene, always there are adjacent to that some consensus sequences. These can be to the left of the gene or these can be to the right of the gene. When towards the left of the gene, that's towards the 5' end, we call them upstream sequences, and when towards the right side of the gene, we call them as downstream. To this upstream, we recognize it as minus, and to this downstream, we recognize it as plus end. Now, see, this is the gene, fine, and now the sigma subunit, it has, for example, I will make it more clear here. In bacteria, we have two promoters, two consensus sequences. One is -10 box. Why -10? We call it as C, because when the RNA polymerase, once it recognizes these promoters, it starts the transcription, and the first nucleotide which is added, we call it as +1. Fine, and that is called as transcription start site. Transcription start site is the site at which first nucleotide has been added. Now, to this plus site, to this left of the plus site, we call it as upstream, and to the right side we call it as downstream. Now see, to this plus site, about 10 nucleotides away from this plus site towards the left, we have a consensus sequence which we call as Prabno box or Tata box. It consists of 6 nucleotides which remain consensus, that is TA, tat, located on the left side about 10 bases away. So this 10, it says that at 10 there's a promoter, there's a consensus sequence which is about 10 nucleotides away from this transcription start site, and minus indicates that it is the upstream sequence. Now, in bacteria there is another promoter which is called as -35 box. So -35 box will be also, it will be 35 nucleotides away from the transcription start site, and minus means it is upstream. It contains a sequence, conserved sequence, which we call as DTGACA, which is located 35 bases away on the left side of transcription site. So in bacteria we have usually two promoters. One is Prabno box, which is called as -10 box or Tata box. It is having a sequence, 6 nucleotide sequence, T88 Tatat, and -35 box. It contains a sequence TTGACA, which is 35 nucleotides away on the left side on the upstream. So in bacteria we are having two main promoters. Usually we are having one upstream downstream promoter also. Leave that. Now we are having two promoters. One is Prabno box and another is TTGACA. It is these sequences which are actually recognized by the sigma subunit, and then the sigma subunit, upon joining here, then the transcription of the gene starts. So a sigma subunit is like, it guides the RNA polymerase that whereas you have to start the transcription, where you have to start the process of RNA synthesis. Without the sigma unit, this core enzyme, this RNA polymerase core enzyme, is not able to bind specifically as to which gene is to be transcribed, and without sigma subunit it will be a wasteful process, because at certain conditions we only require certain genes. So this sigma subunit, it makes it more specific, more precise as to which gene is to be transcribed. With this we have come to the end of this, the basics in transcription. Inshallah, in next lecture we will be studying about its mechanism. Thank you so very much. Start a discussion here. I would love to answer your questions. Please be active here.